Hello and welcome dear viewers to the interesting topic on nitrosamine impurities, and in this video I will walk you through the complex nitrosamine impurities. So I have made some videos on classification of nitrosamines, and other videos are there for controlling the nitrosamines and what are the sources of nitrosamine impurities. So you can watch those videos to have a complete understanding about the nitrosamine impurities. And in the current video, we are going to see what are the complex nitrosamine impurities and how their risk is assessed and how the limits are given for those complex nitrosamine impurities. In short, this video is for how to deal with the complex nitrosamine impurities. And nowadays, nitrosamine is a very important topic in the pharmaceutical field, and many of the organizations are dealing with this type of impurities in their formulations. See, nitrosamines can be divided into two categories like small nitrosamines and the complex nitrosamines. So the generalized structure of the nitrosamine involves the N double bond O, and it is a nitroso type of compound. So based on the chemistry, the nitrosamines may be divided as small nitrosamines and the bigger nitrosamines or complex nitrosamines. So I said the word bigger nitrosamines, it is for only your understanding. You can remember like bigger structure or bigger group or bigger molecular weight nitrosamines and the small nitrosamines. So mostly these nitrosamine originate from the synthesis route, and these small nitrosamines are different type of impurities, and regulatory authorities like USFDA and EMA has given their intake limits. So these are known as AI limit, that is acceptable intake limit, given in nanogram per day. So small nitrosamine impurities are like NDMA, NDEA, NMBA, NMPA, NIPA and NDIPA. Others might be there, but these are the classic examples of small nitrosamines. And the nitrosamines have very stringent intake limit based on the maximum daily dose of the formulation. So from 26.5 nanogram per day to 96 nanogram per day. This is the AI limit. So this is very stringent limit for the nitrosamine impurities. Then coming to the complex nitrosamine impurities. These have bigger structure and their structure resembles to the or is similar to the API molecule. So these are also known as NDSRIs, or these are called as drug substance related impurities. Nitrosamine drug substance related impurities. As these are the nitrosamine impurities and their structure resembles to the drug substance. That's why these are called as NDSRI impurities. Their structural similarity to the API or drug molecule lead to the less potency or less mutagenicity, because the structure of these nitrosamines resemble to the API structure. So it may be said that these have comparatively less potency or less uh, mutagenicity potential. So these typically lack compound specific mutagenicity and carcinogenicity data to inform safety assessments. Nowadays, the regulatory authorities are giving the limit to the specific impurities, and they are keep on updating the limits. So it is very important to check the FDA site or EMA site for specific intake limit updation. The example of complex nitrosamines is like N-nitrosoquinapril, and its API is quinapril. So if quinapril is having a nitroso group, then it will form a impurity N-nitrosoquinapril. Then N-nitrosoorphanadrine. These are the typical examples I have taken. Many you can find from the literature. Then coming to the risk assessment. So whenever you find the nitrosamine uh, impurity alert or impurity potential, always check the API structure. So if there is a presence of amine group, that time it is likely to have the uh, nitrosamine impurity formation, and based on the API structure, a risk can be identified or assessed. So first step in the complex nitrosamines is to have a complete risk assessment for complex nitrosamine or NDSRI formation based on the API structure, that is presence of amine group, then presence of excipient into the formulation which may be contaminated with the nitrite or nitrate impurities like MCC is there, microcrystalline cellulose. Sometimes starches also contain these impurities. Sometimes cross carbamylose sodium is there. So all these excipients are required to be evaluated, and all these excipients are to be taken into a risk assessment consideration. Mainly nowadays microcrystalline cellulose is also available with low nitrite content. So if you have the problem of nitrosamine or nitroso group formation into your API due to presence of these impurities and due to the presence of amine group, then you can use low nitrite grade excipient. Another is the nitrosetting agents. So these may be the acidic agents and nitrosetting agents like nitrite impurities. Then manufacturing process. What type of manufacturing process is being finalized or it is finalized for the formulation, that may have some impact onto the complex nitrosamine formation. So generally wet granulation and the use of high temperature in drying, uh, like fluid bed drying, it favors the NDSRI formation. Then if API is micronized or if it is meal type of API, then there are chances of nitroso group formation and NDSRI formation in the jet milling or in the micronization stage. So this information is required to be collected and studied properly to have a risk assessment in your hand. Now from the risk assessment, you can have two options, or there are two ways. If risk is not there, then there is uh, no requirement of testing the formulation. But if you identify the risk, then the analytical testing will come, and then the impurities are required to be controlled. So then if the risk is there, and if it is found that the impurities may get formed, then the identification of nitrosamine impurities is required, and after identification its mutagenic potential need to be checked. If you come up with a impurity and you have identified that impurity, then its mutagenic potential is required to be studied. So mutagenic potential can be studied by uh, 2-3 ways are there, like SAR studies. So structure activity relationship studies are there, and the databases are available for performing these studies, and these uh, alerts are given based on to the structure of the impurity. So if the structure contains the uh, functional groups or bonds or presence of some items which are prone for the mutagenicity, so based on that structure activity relationship database, the structural alerts can be taken. If the database shows that there is no structural alert for this impurity, then generally it is considered that the risk is mitigated, and then the impurity needs to be controlled, sometimes maybe with the uh, ICH guidelines for the impurities. Like it is not considered as a genotoxic impurity, and based on the ICH limits for general impurities, the based on the MDD, these impurities are controlled. Now, if the structural activity relationship or SAR denotes that it may be a mutagenic impurity, then its mutagenicity confirmation is required. And for confirmation of mutagenicity, first step is to conduct the Ames test, and Ames test will give you the information about the mutagenicity. If Ames test is negative, that means though the SAR database has shown the impurity as mutagenic, but Ames test is negative, so there is no mutagenicity into the compound, and the control strategy as per the MDD and ICH guidelines can be done for generalized impurities. If the Ames test comes positive, then it is considered as a mutagenic impurity, and after the Ames test, to have the limit for the complex nitrosamine, in vivo studies or in vivo toxicity studies are required to be performed, and these in vivo testings are done in the animals. So based on those animal studies and toxicity studies, the limits can be derived for the complex nitrosamines. See, the things to be considered and understood and remembered are that the complex nitrosamines have the bigger structure, and these are different from the small nitrosamines. Generally, the small nitrosamines are much potent, and these all small nitrosamines have the mutagenic potential. But for the complex nitrosamines, there are less chances of the mutagenicity compared to the small nitrosamines. See, always remember that small nitrosamines are more toxic. They have mutagenic potential, they have carcinogenic potential. That means the genotoxic potential is more with the small nitrosamines, but it is not that much with the complex type of nitrosamines. These complex nitrosamines may be less potent than the small nitrosamines, and their biological response may be different, because small nitrosamines may interact with the enzymes and may lead to the DNA mutation by alkylation, but the complex nitrosamines may not behave in the same way. The complex nitrosamines may or may not interact with the enzymes, the same enzymes for which the small nitrosamines get interacted easily. So it is not the same case with the complex nitrosamines. So complex nitrosamines may or may not interact with the enzymes to produce the diazonium ions, which leads to DNA mutation by alkylation. See, to mutate the DNA, formation of diazonium ion is required, which is very common with small nitrosamines. So the complex nitrosamines may interact in a different way, and that's why their mutagenicity may be different or less potent. Then we can use the available data for the complex nitrosamines. If you are working on a generic formulation, so you may have uh, those data available with the agency or with the data standard databases, and you can use that data, and you can give the AI limits once the structure is identified. So you should have the structure, then you can uh, use the available data. If it is not available there, then CPCA approach can be taken. And CPCA approach is the carcinogenic potency categorization approach. So based on the CPCA approach given by the regulatory authorities, the structure of the nitrosamine is studied and the rank is given as per the CPCA. So based on that, the levels of acceptable daily intake ranging from 26.5 nanogram to 1500 nanogram per day can be given. So from here you can understand that for complex nitrosamines you can get the higher intake limit compared to that of the small nitrosamines. So this reference I have taken from US FDA guideline for nitrosamines, and based on the CPCA potency category from 1 to 5, the recommended acceptable intake is given. So for category 1 it is 26.5 nanogram per day, for category 2 it is 100 nanogram per day, for category 3 it is 400 nanogram per day, and for the NDSRI impurities specifically with higher intake, that is category 4 and 5, these type of NDSRI impurities are getting the highest AI, that is acceptable intake per day, that is 1500 nanogram per day. So based on that CPCA approach, a higher limit is given for the NDSRIs. So in this way, the complex nitrosamine impurities can be dealt, and the CPCA approach can be beneficial for the complex nitrosamines. So unlike small nitrosamines, complex nitrosamines may be with less potency, or sometimes these may lack the mutagenicity. If the mutagenicity is not there, if the complex nitrosamine doesn't show any mutagenicity, then as a general impurity, the control strategy can be given. So this is regarding the complex nitrosamines, and I hope I have covered maximum information about how to deal with the complex nitrosamines. So if you have any question, if you have any suggestions, kindly comment it, and if you find this video helpful, please inform me. I will be very happy, and I will try to give you the good information in upcoming videos. Kindly do like, share and subscribe to this channel, and thank you for watching the video.